Swayam Prabha. Digital India. Educated India.
Hi everyone, welcome again to the course of structural biology. We are at the end module where the topic is structure based drug discovery. Though it is called structure based drug discovery, but it is about the general concepts of drug discovery and how the structure solution dynamics help the process. And if you remember, in the last module I have talked about protein engineering, enzyme engineering, and this module drug discovery. These two modules talks about how the things we have learned in the last 10 modules, like the 10 modules, are coming to an application. In protein engineering, in enzyme engineering, we have seen how the world is being opened for applications. Coming here in the drug discovery, we will see the methodologies which are helping us to identify a potent small molecule which could be used as drug. But nowadays the drug molecules are not even, I would say, like limited on the small molecule and its chemistry. We are getting peptide drugs, we are even getting protein drugs. So, the options are opening up, the field is getting expanded. Today I will talk about introduction to structure based drug discovery. First, to come to drug discovery, I want to ask you a question: what do you mean by drug discovery? Because with a concept in your mind, it would be helpful to follow the module, follow the course. I have my own very unique definition of drug discovery which I want to present to you, which is creating small changes in chemistry, and by making that small change there is a huge effect which will be affect the biology. So, small change in chemistry, a big change, a big effect to biology. How or what is the process? So, to explain you what I mean by this, I take the help of a case study of a drug designing, which is the area of my research, and try to explain the whole concept. In my introductory classes, I have already talked about the molecular level of the DNA structure. This is a double stranded DNA. If you look at here, you will see the red molecules, red and yellow, they are phosphate, the bases are hydrogen bonded and they are stacked in a way so that the like strand looks like there are stairs in between. So, if you look at the molecular component, there are three major components you could see in DNA or even in RNA. There is a sugar, in DNA it is called deoxyribose sugar, whereas in RNA it is called ribose sugar. There is a phosphate group. When there is a synthesis of a DNA, it comes with 3 phosphates, is released to phosphate and get the energy to make the polymerization reaction going into thermodynamically favorable direction. Then the third one is the nitrogenous bases, and you know in case of DNA it is AGCT, whereas in RNA the thymine is replaced with uracil. So, if you look at the entire composition, the only difference happen in the nitrogenous bases. So, first take a look at the nitrogenous bases. The nitrogenous bases are of 4 type: adenine, guanine, cytosine and thymine, as I told, but among those 4 they are also divided into 2 classes. Adenine and guanine is called purines. Here there is a 6-membered ring followed by a 5-membered ring, whereas cytosine and thymine, they have only 6-membered ring and they are called pyrimidine. And there is a rule how they perform or they form the base pairs. You could see adenine form hydrogen bond with thymine, where 2 hydrogen bonds are formed, and cytosine with guanine there are 3 hydrogen bonds. You could also see in the animation that adenine, how and what way they are forming 2 hydrogen bonds with thymine, and how cytosine form 3 hydrogen bond with guanine. These 3 not only help them to form the double strands, but also help them in helping in recognition. So, hydrogen bonds are intrinsic characters used in the living organisms to identify A over T and G over C. So, they are very critical. And with that background in mind, now I am going to introduce something which is called nucleoside analogues. So, you know about nucleosides, now I am talking about analogues. What are analogues? If you have a compound A and then you make variations A1, A2, A3, A4, they are called analogues. So, by the definition, nucleoside analogues are variants of these nucleosides. Let us start with a simple one. You see deoxycytidine, you know how it could form. Just now we have seen that deoxyguanosine form 3 hydrogen bond with deoxycytidine. Now, if you see, only this portion is involved in hydrogen bond, correct? So, now if I cleverly change in the other part, in the sugar part, what will happen? Before going in details, I could show you that we designed a compound and did not design, actually get rid of this hydroxyl group. Now, if you see now, this hydroxyl group is doing nothing to recognition hydrogen bond. So, have no effect in GC base recognition, right, which clearly means that dideoxycytidine is, if you see, going to form exact hydrogen bonding pattern like deoxycytidine. Now, why this is very critical? Think about now, because it could have the same hydrogen bonding pattern like deoxycytidine, it could easily insert in the DNA by the process of recognition through GC hydrogen bonding. But once it will insert there, incorporate there, what will happen? Let us see. So, this is a normal DNA synthesis where a A is there, so T is coming, C is there, so G is coming, G is there, so C is coming, and T is there, so A is coming. In that way, a newly formed chain is continuing to form a new DNA molecule, whereas instead of this deoxy, if we have dideoxy, what will happen? Somehow, if a dideoxy could incorporate, it would never allow the DNA to extend further, which will lead to the termination of DNA synthesis. So, this DNA would be stopping its elongation in the process of polymerization. So, when you do not have DNA, your cell could not survive. So, it would lead to cell death. So, you need a compound like this for that, which is coming from the compound we have designed. So, I hope you could understand the process what I talked about. Here we just removed a hydroxyl group from a nucleoside, and that small change leads to DNA termination, followed by further affecting the cell death. One hydroxyl group, a death of a cell, stopping of a life. That is what I call a drug designing. So, this is nucleoside analog, and that is their critical role in biology. So, nucleoside analogues are compounds with identical hydrogen bonding properties, as you have already seen that in the picture where I have put the GC hydrogen bonded pattern and then brought the dideoxy one. So, nucleoside analogues are compounds with identical hydrogen bonding properties of natural nucleosides. When incorporated, they use this property to block the synthesis of a DNA chain, leading to cell death. So, I hope you guys understand the basis of drug discovery, because if you do not understand what is going to happen, you do not understand how the methodologies are developed or what are the new methodologies which should come and incorporate and speed up the process. So, first you should understand the basic, which is the drug designing. Now, see, so this nucleoside analog could cause a cell death. You could use these as a drug. How you could use this as a drug? If a person have cancer, what happens in cancer? Cells divide with uncontrolled condition. So, you have normal cells and you have cancer cells. The cancer cells are growing. Somehow, if you could incorporate this nucleoside analog, they will definitely kill the normal cell, but because the process is speeded up, they will kill more and more cancer cells. And that is the thing you probably have seen when we say that a person is affecting or diagnosed with cancer, getting cancer chemotherapy. In most of the cases, they are treated with nucleoside analogues, which goes to the both the cells, normal cell and cancer cell, kill them, but the strategy is here we take the advantage of a cancer cell, which is the disadvantage. What I mean by that? The cancer cell grow much faster. So, these nucleoside analogues are entering there, stopping DNA synthesis, then killing the cells. So, that tells us that this nucleoside analogues could be working as a drug, as a therapeutics. And when I am telling this, I could also tell you another, just one more thing, more information. If you see people getting cancer chemotherapeutics, you see that they vomit, they become tired, they lose their hair. This is because this nucleoside analog, they kill the cancer cell, but at the same time they are killing the normal cells. So, if you do not want that, no one wants that, no one wants to lose their hair, no one wants to get the cells really get stressed condition, they are killed and all, we do not want. So, we want specific therapeutics, and for that you have to work on this compound and improve the molecule. That is the process of drug optimization. So, you would understand a lot of things. First, you understand what is drug discovery. Drug discovery is changing small in the chemistry, making big in the biology, which is clear here. Then you would strategize this to use as a drug, but this drug is not optimized because in addition to killing the cancer cell which you want, it is killing your own cells also, which we do not want. So, we have to go through the next cycles. So, here this molecule would be considered as lead molecule, and then further level of drug discovery would be going on to make the drug better. So, this is a classic example of small change in chemistry as the hydroxyl group is modified here and affect biology greatly as this small change leads to cell death. Idea of other drug designing from here, as I told you, optimize, you want to make it a better drug, so that non-specific, or literally to say that the killing of normal cells would not happen, you want to work more on this drug moiety. So, nucleoside analogues, if I am talking about a dideoxycytidine, dideoxyuracil, dideoxythiamine, the case is pretty clear and pretty easy to understand for you. Okay, this does not have this hydroxyl group. So, once it would introduce to the DNA polymerase would be unable to continue, right. But then I will introduce new molecules, 3TC, which is lamivudine, this is troxacytidine, this is dideoxycytidine, this is D, this is L. So, there are 3 nucleoside analogues where the sugar, here the sugar lose the hydroxyl group, but here the sugar is having L chirality instead of normal D. So, how it should work? That was a enigma for a long time, though in reality as a drug, lamivudine, troxacytidine, L-deoxycytidine, they all are working much better than those straightforward analogues. They had little bit more, I should not say specificity, I would rather say less non-specificity. So, they are killing the natural cells at a speed much less than what these dideoxy compounds are doing. But the mechanism was in question, and in my PhD I got opportunity to work with this. So, what we did, we crystallize all of those compounds with a target enzyme called deoxycytidine kinase, and we have clearly shown the molecular details of how these molecules are actually interacting with the enzyme and how the catalytic residue GLU53 is coming close to the sugar where it like position where it have to hydroxylate. And the most interesting part of this designing is not this. If you see the comparison between L-deoxy and D-deoxycytidine, you will clearly see that in case of D-deoxycytidine it is here, whereas there is a huge puckering, sugar puckering happened, and this sugar puckering helps the L nucleoside to present its atoms close to the GLU53 as the D-deoxycytidine did. So, if you do not understand, I repeat, all those D and L nucleoside analogues have to be phosphorylated by this enzyme which is called DCK, deoxycytidine kinase. Here when you see them, you just see, you will realize the fact that this GLU53 is near to this, but if you understand now, so this hydroxyl group could be phosphorylated by this enzyme. But the more surprising or more exciting fact would be coming to the comparison of L-deoxycytidine and D-deoxycytidine, as it is actually comparison to apple. This is clearly showing that there is a huge rotation here, and this huge rotation ultimately brought both the hydroxyl group in nearly same position. So, be it L, be it D, you could say like this is D. So, this is presenting, this is L, it somehow present it like that. So, that is what the mechanism is talking about. And as I was talking about how drug designing evolute, we talk about this is dideoxy, the concept is direct and strategy is straightforward, that the absence of this hydroxyl group do not allow the DNA polymerase to elongate the ring. But when you come to the L nucleosides, you do not know what is happening actually. What is happening here is, if you see, the L sugar is rotating slowly and moving the hydroxyl groups away from the enzyme, from the DNA polymerase. So, it is little bit complicated, but the research comes up with the truth. It shows even if it have a hydroxyl group, it would not present that to the enzyme L DNA polymerase to further elongate. And then coming to the third type of molecule, which is wonderful antiviral, specifically working against herpes simplex virus. This is called acyclovir, ganciclovir, phencyclovir. They are a series where the sugar molecule, the chain, the base is taken out. So, instead of being a closed ring, this is a open chain. So, this open chain is even more interesting mechanism, how it actually very specifically kills the herpes virus. So, herpes virus transmits sexual disease and it was a terror. Now, thanks to those Vir drugs, acyclovir, ganciclovir and all, you take one tablet and the virus would be totally killed. So, this is one series by which I could present you a lot of things, how we could understand drug discovery, how we see that there is evolution of a compound getting from nucleosides, how you could modify and get better drugs, and how the drug optimization cycle continues with targets 1, potency of the drug, 2, less to less toxicity of the drug. So, if you follow me, you will now have questions, you have get few answered. What is the process of drug discovery? I have already shown you what is drug discovery, but how to do it, what are the processes, it should be in your mind. Then as I told, this is a structural biology course. So, our drug discovery would be also, in addition to other methods, we have a major focus on structure based drug discovery. So, how structure based drug discovery, where the structural information is used, when it is not structure based drug discovery, then what happened differently, all of them are questions. As I told, how it is different from ligand based drug discovery. So, if you now understand, in most of the drug designing there is a protein and a small molecule. The small molecule is called ligand and this is called receptor or biological macromolecule. When we have to use these, we need its structure in high resolution, and when we use those information, it is called structure based drug discovery. When we use this information, the information of the small molecules, it is called ligand based drug discovery. Remember, peptides also come under ligand based drug discovery. What do you mean by lead compound? So, if you remember, the dideoxy you have initially started, then make modification. So, the first one which gives us the drug like activity is called the lead compound. What are the challenges in the field of drug discovery? We will go through and discuss one after one the challenges and how they are called drugs. So, what I mean by this? You have a small molecule, okay, or a peptide. When you say, or when you have the confidence to say that this small molecule or this peptide are drug, I want to explain my thought with one example. Suppose we have a small molecule. That small molecule could kill a mice. So, you could take the bread, you could put the jam and you put that small molecule there, you call it poison, you keep it there, the mice will come, eat that and that will kill the mice. Is this a drug? Apparently yes, but think about, if you are infected by corona, can you use that against corona? No. Why? Because this compound would kill you. So, a compound killing an organism is not considered as drug. a small molecule or a peptide could be considered as a drug only when by consuming this it would not have lots of side effect to human. So, we have to look at the druggability of a small molecule. How we will check the druggability? We will see that too. I talked about lead compound. A lead compound is a small molecule that serve as the starting point of an optimization involving many small molecules that are closely related in structure to the lead compound. Many organizations maintain databases of chemical compounds. Some of these are publicly accessible, others are proprietary databases contain an extremely large number of compounds. The most popular ones which are accessible are ACS database, which contain 10 million compounds. There is database called Zinc. When I will go to the structure based ligand based drug designing, I will give you a table of the popular databases of small molecules. So, coming back to drug discovery, I hope you could understand my unique definition of drug discovery. From there, let us proceed. Drug discovery is the process by which new candidate medications are discovered. Considering history and how the process organized, we could divide the process of drug discovery into two major parts. One is traditional drug discovery and another is rational or logical drug discovery. So, what you see now the pharmaceutical companies are doing are rational or logical drug discovery, but when it started we mostly did traditional drug discovery. It is not that traditional drug discovery did not have any logic. We know about Chinese traditional medicine practice, we know about Ayurveda, we know about alchemy in Arabian society. They had their logics, but it was not well documented, it was not proper scientific procedures, at least in our eye. So, traditional drug discovery. The traditional drug discovery involves the origin of drug discovery that evolved in natural sources, accidental events, etc. It was not target based and not much systemized or logical like current days. Introduction and popularization of next generation sequencing, the NGS we talked about, better structure solution techniques, we discussed them in detail, introduction of machine learning based prediction along with improvement and advancement in pharmaceutical science and technology made it revolutionized to much more organized and system oriented modern drug discovery, which is not traditional drug discovery. You could divide traditional drug discovery majorly into 5 topics. 1. Random screening. 2. Trial and error method. C. Ethnopharmacology approach. D. Classical pharmacology. E. By serendipitous discovery. I will try to introduce them, but I will introduce them briefly. You could talk more and more about all of them, but as you know, this is only a module where I have to talk about. So, I just spent one slide for each of them to introduce these things. Random screening. Random screening involved a collection of synthetic compounds, chemicals, natural products which could be assayed by experimental assay method. So, suppose you want to develop a drug against diarrhea and you have a assay system where by adding the drug you could differentiate between a small molecule which is affecting in a good manner or which is not affecting. It involves two major approaches. One, screening of selected class of compounds like alkaloid, flavonoids, any synthetic library collection, or screening of randomly selected traditionally identified medicinal plants. In traditional drug designing, medicinal plant have a huge role. As we know from ancient Indian civilization, they used to culture the process called Ayurveda. Ayurveda depends on several plant products based on 1000 years of experience from the saints who used to live in forest and intensely follow the animals or birds. So, they live with the animals, with the birds. So, how or what are the process the animals are applying when they have any problem, they have experienced them, then they did some experimentation, and all those are passing from one generation to next generation. That is what the culture of Ayurveda. Coming to trial and error method. Trial and error includes berries, roots, leaves and barks that could be used for medicinal purposes. Few examples are cinchona bark contain quinine, which is used to reduce fever in malaria. Leaves of Azadirachta indica, which is neem, are used as anti-inflammatory and antibacterial properties. Licorice roots are traditionally used to treat stomach ulcers, bronchitis and sore throats. So, in that way you have collection of plants, you apply, you see it is affecting, okay, this is right. So, that is called trial and error method. Ethnopharmacology approach. Ethnopharmacology is the study of medicinal plants used in specific cultural groups. It involves the observation, description and experimental investigation of indigenous drugs. So, this is traditional, as I told, Ayurveda, Chinese traditional, African traditional medicinal practice. So, it is based on botany, chemistry, biochemistry, pharmacology and many other disciplines like anthropology, archaeology and history. Andrographis paniculata, which we know in India as Kalmeg, was used for dysentery in ethnomedicine, and the compounds responsible for the activity were found to be andrographolides. Classical pharmacology. It is alternatively known as function based approach. Without the prior identification of drug target, anciently drug discovery process were often based on measuring a complex response in vivo, such as prevention of experimentally induced seizure, lowering of blood sugar, suppression of inflammatory response. One very good example is the discovery of foxglove in Europe, which used for drug preparation that contain cardiac glycosides, particularly one which used to get in high amount is digoxin extracted from various plants of this genus. This is Digitalis purpurea, which is common foxglove. The end one is the serendipitous discovery, and I will make it a story which I would follow for further case studies also, because there are many different type of diseases, many different type of systems. So, if I start talking about each and every system, you will all be confused. So, what I take is what I know is about antibacterial. So, bacteria was first identified in the 1670s by van Leeuwenhoek. I have already told you, following his invention of the microscope. The relationship between bacteria and diseases gradually set up in the 19th century. Since then, researchers started to try and find effective antibacterial agents. Though there were local usage of traditional material against microorganisms like Ayurveda in India, traditional Chinese medicine in China, alchemy in Arabian countries, but there was lack of existence of any global medicine. And the concern was somehow solved by a discovery in 1928 by Alexander Fleming. He was a doctor who was working on Staphylococcus, and once he went to a vacation, you could see the plate. This is a bacterial Petri plate, where the colonies you see here, which is written normal bacterial colony, are Staphylococcus colony, they are already grown. When he came back from vacation, to his surprise, he realized that there is a mold grown in the Petri plate, and very interestingly, the region where the mold grown, the bacteria is not present there, they are already killed, the colonies are gone or the colonies are minimized. After some following up experiment, what he found, I definitely want to use his experience. When I woke up just after dawn on September 28, 1928, I certainly did not plan to revolutionize all medicine by discovering the world's first antibiotic or bacteria killer, but I guess that was exactly what I did. So, in one moment, the worry, the lack of communication around the world was solved, because somehow the world got a beautiful answer, the magic bullet which could kill any and every bacteria. Is it good for us? I would like to discuss it in the next classes, but now I will tell you about the success story following up the invention of this penicillin. So, penicillin had a wonderful time to launch. It had a beautiful sense of debate. Why? Because when penicillin was invented, World War I was already finished and World War II is coming. If you see, in World War I, around 18% of the soldiers, it is a huge amount, 18% of the soldiers were not killed in the war. They had minor wound, but that wound leads to bacterial infection following up the killing of the soldier. 18% of the soldiers killed there. And Second World War, which is more fierce in terms of the intensity, only less than 1% people were infected or killed thanks to penicillin. And you would be more surprised to know that that time penicillin was not even a drug. It was the mold, and the mold is put under a squeezer and squeeze, and the juice coming out from the mold is actually allowed to put on the wound, and that was good enough to save them. Suddenly all the countries who were involved in the war get the utility of penicillin, and penicillin was accepted globally. In 1945, Fleming, Flore and Chen were awarded Nobel Prize. So, 1928, 1945. You understand already why Fleming, because he invented penicillin. Why Flore and Chen? Because they were in the University of Oxford. They had done the actual molecular level research and come up the structure, the typical beta lactam ring, and all those molecular characterization was performed by them. So, these three got Nobel Prize, no problem. The problem is, in his acceptance speech, Fleming presciently warned the overuse of penicillin might lead to bacterial resistance. First time the world is listened to the term bacterial resistance. Today you look, the entire world, a burning problem is bacterial resistance. Alexander Fleming had identified it, understood that, and that is the reason from 1928 to 45 no further work, means innovation regarding penicillin comes from Fleming. Imagine you are the first antibiotic provider. Any and everyone would want to utilize it, further write grants and do more work. Alexander Fleming did not. And why he had answered it in 1945, today under 2021 standing here we are realizing what he told, but that time no one listened to him. Why? Answer is there. Beta lactam, this 4-membered ring, brought so much money, as I told, because of the excellent timing of launching penicillin. Before the World War II, a lot of soldiers, they saved, their life was saved, and all of them suddenly become vocal about penicillin. It became globally accepted drug. Utilize that craze in terms of money. A lot of companies which probably you all heard about, Merck, SmithKline Beecham, Danipon, Sumitomo Pharma, Novartis, all of them started with a derivative of penicillin. So, you see all the famous companies, you go back, you see that they start having a patent of a beta lactam. It would not be exaggerated if you could realize the fact that the concept of pharmaceutical industry started with the innovation of penicillin. It also stated that time people invested $1 in beta lactam oriented pharmaceutical market got minimum refund of $100, a minimum. So, in one side it is a huge money, in other side this was helping to set up industry. Means if you see the same time when people are becoming aware, people do not want to work under other people. So, in the world there are always two type of people, one who like to order and one who obey order, and this is the majority. When they start understanding the model, these guys come up with a different model. So, when farming under some people was understood by majority not working as a good model, people choose this pharmaceutical industry along with other industries as an alternative. So, combining power and money, nobody listened to Fleming, but that is not the topic of today's class. Today's class, okay, so they have developed pharmaceutical industry, they earn a lot. What happened? And if you see Bollywood movie, especially in 90s, we always end up in a happy moment. We always say they lived happily ever after. So, the newly formed company started well and went through the golden era from 1940 to 1970. They start realizing that one or few products they are banking is not enough to run a company forever. So, they start investing into new research and development. They include life scientists and chemists to bring new targets, new drugs, so that they could patent and continue the industry. But they face challenges. Before going into challenges, let us look at the traditional drug designing. You have this lead generation, natural ligand, you do screening, then you get the biochemical testing. If you get positive result in biochemical testing, then you go to biological testing. When you get success, you do the synthesis of new compound in one side, which is a drug design cycle, whereas in the other side, if it gives promising result, you go to the preclinical studies, clinical studies. So, what involves traditional drug designing? Lead generation, biochemical testing, biological testing, synthesis of small molecules, peptides, preclinical studies, and if that would work, clinical study. So, the challenges are the process of bringing a potent drug to market needs a huge investment in time and money. The Tufts Center for the Study of Drug Development estimated that it needs incredible $2.6 billion to develop and bring a drug to the market. The cost has increased almost 150% in the last decade, making it more problematic. At the same time, the failure rate is also increased to nearly 90%. This means that almost 70% funds are costed on account of failure to pass through clinical trials due to a lack of efficacy or adverse side effect on the preclinical or clinical trials. Whereas, bringing a pharmaceutical drug to the market takes 10 to 15 years on an average. There are definitely advancement of combinatorial chemistry, high throughput assays, which definitely enhance the process, speed up the process, but incorporation of those advanced techniques are still time consuming and very expensive. On the other hand, this time and cost enhancement push the company to increase the price of the marketed drugs, but due to intense competition and limitation to affordability of common consumers, if you increase the price of the drugs including the R&D which you need, the company needs to cover up, but as a common consumer the price of the drug enhanced to a level so that we cannot afford it. Also, there are competition, so one company cannot suddenly enhance the price, then they will be not getting proper cell targets. So, what is the way out? The limiting factors as it was looking in normal eye is you have to expedite the process of drug development. You have to make the process in more cost efficient way, and you have to minimize the failure rate. So, all three you know, but not easy to do. Expedite the whole entire process, make the process more cost efficient and minimize the failure rate. And when I am talking about or discussing about that, remember very well, we are not discussing the problem of drug resistance. I will discuss in a case study at the end specifically including it, but now I just discuss normal drug designing for the sake of simplicity of the model discussion. So, the new strategy is to take the traditional method which are converting to high throughput, replacing it somehow by computational method, computer, to get to the achievement, the dream goal, the lead development, replacing the experimental way as much as possible using computer. So, for that we need good logical library generation, high resolution structure, computer algorithm, and incorporate the modern technique of machine learning. And that brings us into rational drug discovery. Rational drug discovery can be broadly divided into two major groups: development of molecules with desired properties for targets having known structure and function, development of molecules with predefined properties for targets whose structural information may be or may not be known. If not known, the unknown target information can be found by global gene expression data. So, the two new approaches, one is ligand based drug discovery, another is structure based drug discovery. The ligand based drug discovery, the 3D structure of the biological target, the macromolecule is unknown, and a set of geometric rules and or physical chemical properties pharmacophore model obtained by QSAR, quantitative structure activity relationship studies, are used to screen the library. In structure based, it involves molecular interaction calculation between each molecule to be tested and the biological target, which usually be a protein. To evaluate the affinity, a scoring function is applied. The 3D structure of the target must be known. So, methods of rational drug discovery, as I told, it would be more computational involvement. Ligand based drug discovery, where it involves QSAR modeling, pharmacophore development, whereas in structure based drug designing it has virtual screening and de novo drug designing. So, what is rational drug designing? Let us take an example using cimetidine, which is also called Tagamet. So, it starts with a validated biological target and ends up with a drug that optimally interacts with the target and triggers the desired biological action. So, you should have a valid biological target, be its structure or not structure, and then using that target you get a drug that interact very well with the target and give you the desired biological factor. So, we are talking about a problem where histamine triggers release of stomach acid. Want a histamine antagonist to prevent stomach acid release by histamine, validated biological target. So, we take histamine and then we look at the molecule and want to make the histamine analogues. Now we get N alpha guanyl histamine, which is already a good analog having desired biological effect. So, histamine analogues were synthesized with systematically varied structure, the chemical modification, and then it screened. In guanyl histamine showed some antagonist property, become the lead compound. So, this became the lead compound. Now, we continue, we make chemical modification of the lead compound, which we call lead optimization. So, from N guanyl histamine we make the modifications, which is burimamide and thiaburimamide. Then we make more potent and orally active, but we include thiourea because of the presence of sulfur. It found toxic. Like metiamide, these have better effect than the non-sulfur analogues, but if you see there are sulfurs and people are commonly allergic to sulfur. So, that is the problem. Replacement of the group lead to an effective and well tolerated product. So, one sulfur is replaced by a chemical group, gives the drug cimetidine, and eventually replaced by Jantac with an improved safety profile. So, the azo one also replaced and we get Jantac or ranitidine with a safety profile. The company Glaxo is marketing it. So, that is called the rational or logical drug designing. You do something, you get something that is having an effect, then you make modification, then you test. So, you apply logic stepwise. The rational approach, the first generation rational approach in 1970, the medicinal chemists considered molecule as topological entities in 2 dimension with associated chemical properties. That was the first time. QSAR concept became quite popular and it was implemented in computers and constituted first generation rational approach to drug designing. Second generation, the acceptance by medicinal chemists of molecular modeling was favored by the fact that QSAR was now supplemented by 3D visualization, because from 2D to 3D you get a lot of effects, especially in terms of spherical effect or interactions which you do not realize in 2D. The lock and key complementarity is actually supported by 3D model. Computer aided molecular design or CAMD is expected to contribute to intelligent lead. Dynamic flexibility concept was started to contribute with the advent of molecular dynamic simulation technique. The third generation rational drug designing, most rational approach employed today. Disease process is understood at molecular level. So, we are not only looking at the target drug, we are also understanding the opposite part, the main biological entity, that is the key to success. So, disease process is understood at molecular level and targets are well defined. Drug can then be designed to effectively bind these targets and disrupt this disease process. Very complex and intellectual approach and therefore require detailed knowledge and information retrieval. CADD, so computer aided drug designing. When you say structure aided drug designing or structure based drug discovery, you cannot put computer aside, or because you get the high resolution structure, you utilize it to develop more potent ones using computer. Drug receptor interaction is not merely a lock and key interaction, but a dynamic and energetically favorable one. So, what It started in the second generation, in third generation this is coming with more modified methodology, more in detail understanding, because mostly we looked at the protein as a static structure, but now we see dynamicity here. So, there is evolution of drug designing. In ancient time, as we have told, natural products with biological activity used as drug. Chemical era, synthetic organic compounds, alchemists in medieval centuries, they are coming with very good synthesis protocols and novel compounds, rationalizing design process, structure activity relationship and computational chemistry based drugs. Biochemical era to elucidate biochemical pathways and macromolecular structures as target as well as drug. Post genomics era, after human genome have been sequenced. Modern machine learning assisted prediction, predictions are much more accurate, training based upgradation is possible. So, NGS is now providing us with new targets. We had the total genome, but now we look into the druggable genes. So, the NGS is not only giving us the genome sequence, it also gives us the transcriptome, the proteome, the condition, how a genome is affected with the change of conditions, how a disease pupil's protein or disease pupil gene is differently behaving comparing to a normal healthy person. All those are giving us new concept in the drug designing era. So, it needs definitely the functional integration of biology, chemistry, screening technology and mathematics, but it needs more importantly the introduction of computer like modeling, like ligand protein interaction, like novel library generation, like ADMET modeling. ADMET is very important in vivo, but in silico ADMET could have saved a lot of money, and then computation based assay would speed up and would further reduce the cost of the process. So, with that I will finish the introductory class. In the next class we will talk about the processes in details. Thank you very much.